Yeah, good evening once again. My name is Leke Aiyesa, and from the University of Göttingen, Germany. And today I'll be talking about genomic prediction at the root, from one plant to better populations. Yeah, the topic is might be a bit misleading, but the idea is how to implement genomic prediction at early breeding stages using the information you have from individual plants to make selection. So what we are used to in breeding research is that we work more with inbred lines and their progenies. And based on this, we use plot-based estimate where we use mean of many individuals that makes this genotype, or we use the sum of their estimate, and this has been the practice over the years. However, with this, we know developing this panel takes time, and when you want to evaluate large number of candidates, you need more space, and also the genetic diversity is quite restricted to the diversity and the number of parental lines you are using. In class, just to be a bit more practical, so you have this simple breeding scheme, and you, at the early stages, say F2, F3, you are making selection based on the phenotypes of the individual plant. And we know that we have a lot of noise at this level that you are not able to ascertain the true phenotypic value of the individual. And so breeders would normally select the individuals into rows to examine and evaluate, and from there to smaller plots and bigger plots. And doing this takes time, yes, but what is the good part of it is that they are able to limit the number of candidates to a manageable size, and also gain more information, especially phenotypic information of the genotypes they are interested in. However, the game-changing idea here is that earlier on we talked about genomic era, changing a lot about our perspective to breeding. And that is that if we consider the declining cost of whole genome sequencing, and we can generate as much information of these individuals at this level, then we can drastically, make selection more precisely, more accurately, and skip the serial evaluation we make that takes this time. So, and this is the angle we are coming from. So we are focusing on using genotyping these individuals, even though we know they have not so perfect phenotypic data, but we want to see how much information we can gain to make selection using such kind of data. And a preliminary experiment by one of the students, just to give an idea of the kind of accuracy of phenotypic value of these individuals. So is to use 8 cultivars evaluated in pure stands and also in mixed stands. And in pure stands, so then for plant height, you would see the correlation between the mean estimate of plant height for the pure stands and the mixed stands. And this is quite impressive, 0.8. So we are not so much losing. So that would mean that if you have a plant of 200 centimeter high, and in the pure stand, in the mixed stand, you have it as 210 or 215 centimeter. So is this so we are not so much losing we are not losing so much information, and that is why we were motivated to try this approach out. So instead of plot-based estimates, we focus on individual estimates, and here we don't have replicates, yes, because an individual is a unique genotype. But this approach is rapid, cost-effective, because you save a lot from further experiments, and also quite flexible. And you can evaluate large number of genotypes, and this can increase genetic gain, not just in the direction of time, but also with selection intensity. You are flexible with how you can select maybe 1 out of 1 million, because you have huge number of candidates. So one of the studies that led to this, a previous studies on this is my GWAS study earlier this year was published, where we used this data set to train GWAS model, of course, and found interesting genes, and also discovered that sometimes you have individual plant-specific alleles which if you have done a bulk genotyping, you would have lost such, possibly lost such allelic combinations in your population. So this is quite interesting to see, and we are motivated to further these studies into training prediction model with such kind of data set. And these are the key questions, just to be a bit focused for this talk, is to see how we can, yes, in the classical approach, you make selection based on phenotypes, and you work more with simple traits at the beginning of your breeding program. Then you begin to add more complex traits in your selection scheme. But here we want to see how we can predict complex traits at the early stage using this individual plant train model. So we will use the data to train the prediction models, and also we want to see how this model performs in different breeding scenarios. So when you have data coming from another year of population or location. But before I delve into the result, this is just an overview of the study design. So we collected some material, landrace material for maize from the German gene bank. So just to have a segregating population, we split the seeds into two-year experiment, and we generated phenotype and genotype data. Some of this analysis are some previous studies we've done, but today we'll be focusing more on the prediction aspect. And this is the experiment design, where we also incorporate some border checks. We've tried some different kind of experiments to improve, to reduce, for instance, plant-to-plant interaction, and also border effect on the phenotypes of these individuals, so we can get the best possible estimate of their phenotype from the field experiment. So in total, we have 1,064 individual plants, and from 50 European maize populations, I'll be showing the result for 12 traits, and we also use shotgun genotyping by sequencing approach to generate our genetic data. So this is how the data set looks like for this kind of population. So you have the broad phenotypic variation within population and across population, and you also have quite prominent genetic structure in this data set. And these data structures, so before I move into the next slide, I will quickly want to talk about genomic heritabilities, because this also we want to know, especially for novel traits or quite difficult traits, we want to know how heritable these traits are using this kind of data set, but we don't have replicates like you would have in the classical approach. So here we adopted the genomic heritability approach, which is common to human genetics and animal-based, human genetics especially. And here we saw that, okay, we have the same trend in terms of pattern of the heritability results, such that traits you have in your classical approach with higher heritability are also showing high heritability, genomic heritability here, and the ones with very low heritability, like grain yield, are also having similar pattern here. So this is quite comforting to see that we are having a data set that is looking very good. And also we discovered that because SNP-based heritability also has been seen to have a lot of missing heritability. So we tried to see how much we can gain and incorporated the epistasis effect, and we were able to improve the heritability estimates significantly by 10%. To answer the first question of how predictable complex traits are for individual plants, and based on the data structure, so we examined 8 prediction models, 6 statistical models and 2 machine learning models, and also some specific models like the extended G-block model to capture epistatic effect, and also this kind of model we use to incorporate the population structure information into the prediction model. And this is an overview of the prediction result, showing the mean prediction ability across the trait. And interesting to see that we have quite high prediction ability for vegetative traits especially, and even for grain yield, we still have very convincing prediction ability, and that is also similar to the trends and what you have in the normal plot-based and panel-based studies. But in this case, one of the things we saw from the model performance, of course, the machine learning model and the statistical model were performing much better here than the blob model, and the extended G-blob model couldn't use the stasis information to improve the prediction results significantly. And to just zoom into a few traits and see what the result looks like, you will see that, okay, we have quite less error also from this estimate, and that green GDS is having the least. But the big takeaway here is that we can train prediction models with individual plant phenotypes, genotypes, and we can still get significant prediction abilities. And we can also apply this to complex traits at the outset of our breeding scheme. So to answer the second question is, trying this prediction, this individual plant trained model on different breeding cases, and that is what we are trying to do here. We have the across. This seems to be the standard, the benchmarking approach scenario where we pull all the data sets from the 2 years and the 50 population and assign them randomly to both the training and the testing set. And we have the live 1 year out where we train with 1 year and predict the other year. And also this local means we have genetic clusters. I showed us how diverse this population has. So we have some cluster information that we basically use 6 genetic clusters. So here we try to see how genetic distance is impacting prediction ability. And the next is using assigning populations to 5 groups. So we have 50 populations, we assign them into 5 groups, which now means mega populations. So we train with 4 and test with 1 mega population. So this is a random assignment. And the last one would be that we train with we hold 1 population out and train with other populations and predict the one held out. So overview of of the results shows that yes, across is fine. We have been seeing the results based on across from the previous slides. But here we also see that the live 1 year out was quite interesting to see that it's gaining some momentum in terms of prediction results, leveraging with the across. And the other part, which is more of the population, the genetic, it shows more about the genetic relationship and how it impacts the prediction result. And for most traits, you would see that the result was not so interesting, aside for local, which is because of the genetic relatedness. And you would see this more clearly here in this slide, where you see that some clusters or some genetic groups are well predicted, or some populations here are also having good prediction results, and some performs poorly. So this, to some extent, shows that how much we can apply our individual plant trained prediction model. And with this, I would come to the concluding part to show that, okay, we are demonstrating a proof of concept that we can use individual plants from heterozygous individual plants from segregating population to train prediction model to enhance early generation selection. And also this method is quite applicable to not just to maize, but we can try this in other heterozygous species like trig, and even in non-crop species too. And that would bring me to the end. These are my supervisors and the division I work with, and also the people I collaborate with. Thank you for your attention.